Funding for this podcast is provided in part by the IPM Network through the USDA National Institute of Food and Agriculture and by various commodity groups through checkoff dollars, including the United Soybean Board, National Corn Growers Association, and Cotton Incorporated.
The I See Dead Plants podcast shares the stories of people and plants, pests and pathogens, and the conflicts among them. Join us as we speak to the folks who attempt to solve complex problems with plants through pest management research. I See Dead Plants is an extension-based podcast created by the Crop Protection Network and the North Central Integrated Pest Management Center and hosted by Ed Zworski.
Welcome back to the I See Dead Plants podcast. As always, I'm your host, Ed Zworski. Today, I got a special treat for you folks. We happen to have a seminar speaker here at Iowa State coming all the way from the UK. Sebastian Eves-van Akker. So we're going to be kind of talking about, in general, some advanced tools for screening plant parasitic nematodes. So, Sebastian, I'm just going to give a little bit of background information on you for people to kind of get to know you a little bit. He got his bachelor's in science and biology at the University of Leeds, went on to get his PhD in plant nematode effectors at the University of Leeds and James Hutton Institute. And he's currently a fellow at King's College at Cambridge, right? So, Sebastian, I like to ask some questions of you to kind of get to know you a little bit better, introduce you even further to our audience. So, how did you become interested in plant sciences?
Well, first, thanks for having me.
Yeah,
really
absolutely.
Appreciate it. And this is a nice opportunity. It's not something we normally get to do, and it's kind of cool to be able to do it alongside like the seminar. So how did I get interested in plants? I don't know, actually. I was always interested in plants. I remember growing up, we had a greenhouse in the garden, and I was like always growing stuff. Yeah. And I remember, so both of my parents were academics, and so the idea of doing research as a job, which is normally quite alien to people, was sort of reasonably intuitive.
Yeah. You were familiar with it.
Exactly.
You were around it.
Yeah, exactly. And so it was just sort of one of these like obvious things to do. And, at the time, the kind of school I went to, everyone went to university and that's just what you did. So I did that too. And I thought, the thing I like the most is plants. Yeah. And so I'm going to go and do that. Now, unfortunately, I didn't have super good grades in school. Yeah. So I couldn't get into the kind of university that would have like a pure plant science degree. Sure. And the closest thing I could find was this biology in University of Leeds, and so I basically picked that and then picked as many plant modules as I could. Yeah, and kind of struggled through the bats and the bees and the all the other stuff. Did what I could. Yeah.
Huh. So you said you had a greenhouse growing up. Like, what types of plants were you working with? Or?
Nothing exotic. Just like tomatoes and like broccoli and the usual kind of
usual garden plants.
Yeah, and the usual suspects. Yeah.
Gotcha.
Yeah, I wish it was something. There's someone in the lab right now who has this incredible collection of citrus plants. Oh. Exactly. That's a bit more kind of exotic, but I was just pretty basic stuff.
Sometimes that's all it takes is some basics.
And I think your question was how did you get interested in it? And I don't know, right?
Yeah.
I think when you look back, sometimes you kind of try and rationalize why you like something. And it's like, it is like why is it your favorite color?
Yeah.
You do not have a good answer.
No.
Right?
No.
But yeah, I can rationalize. And I think, there are some things about plants that I like, so I like the fact that they have this like developmental plan that is plastic and this is cool. And then from a science point of view, there are things you can do with plants that you just can't do with other organisms, right? So,
Well, that's funny that you say that because essentially I was on a track to be pre-veterinarian when I found that I was interested in plants and, plants are much easier to work with in certain senses, right?
In many ways.
Yeah. So how about nematodes? Where did you find the nematode?
Yeah, exactly. So final year project. I don't know what the equivalent would be here, but, undergraduate, degree, we have these, I think it's like 6 weeks or something like that, that you would spend in the lab. And you pick one of the lab or one of the faculty in the department and you do a lab project. And I picked one lab. It turned out to be one of the only of 2 plant nematode labs in the whole of the UK. And now there are 3, right? So it's not like there's a lot to choose from, right? So it's just totally I don't know if serendipity is the right word, but totally wandered into it randomly, and really liked it. So they're just really interesting. And I think I'm kind of the kind of person that the more I get into something, the more interesting it gets. Right. And then and then I look at it more, and then the more interesting it gets. And so I do feel like that this was the moment when my career was decided, whatever I had worked on, whatever was that first project, the first proper exposure was probably what I would end up doing now. Because, basically what we do now is almost exactly what we thought about then, but we can address the questions in different ways. And
yeah, that is that thing that makes the first impression on you.
Yeah.
That probably goes back into like, why are you interested in plants? It made some impression on you, and
it's just cool. I just like them.
I guess at this point, let's talk about some of the work that you do. I mean, I've talked about nematodes a fair bit on this podcast, so I think that longtime listeners especially, know at least a little bit about nematodes. But as far as nematodes go, I want to know for our listeners, what types of systems do you work in? What types of nematodes do you work with in your lab?
Yeah, so like tiny minority of nematodes, right? So, most nematodes, totally good guys, benign. You've probably heard this before. Dominant animal on the planet kind of thing, right? And so, there's a subset that are parasites. And, for me, those are kind of interesting because of the kind of interactions they have with hosts. And so we work on the group of parasites, but of course, as you go down, you can split this in different ways. This parasitism has evolved multiple times, and there's different kind of groups. And we work on the major group in the UK, which is called the cyst nematode. So I don't know if you've had this on the show before, but this is a kind of nematode that lives in the soil, and it can remain dormant in the cyst in the soil for a long time. So it could be decades, something like that. And then a plant grows nearby, and the nematode hatches, and it migrates toward the host, infects the root. And then for me, the thing that captivated me is that it then makes the plant make a new organ, right? And that is totally crazy. So, one of the ways I like to explain this is if you imagine that you got infected with a disease, okay, and it doesn't matter what the disease is, it could be anything. And then, but it made you make a new organ, okay? So not like another organ you already have, not another kidney, but you got a new thing, right? And so this new thing it uses your genes, right? So all of your developmental program, but it's just rewired in a different way that is new. And the sole, as far as we're aware, the sole, reason that organ exists is to feed the nematode. And that's wild, right? And so obviously, in doing this, they drain the plant resource, it feeds the nematode, and that's how it contributes to the, to the crop losses. But, it's really it's the interaction between the plant animal kingdom and this kind of development story that captures me, I think.
Already, we could wrap it up and I could be happy with that. I mean, I worked with cyst nematodes for my master's. I don't think I've ever heard anybody explain it quite like that. I've never really thought about it in the sense that, maybe there's something, but I'm not aware if there is in a human being where a pathogen causes it to create a unique organ in its body. That's such I had never thought about it like that, how unique it is.
I mean, part of that is because of plants. So, plants have plastic development. They do that all the time, which means that they, their body plan is not determined at birth or, genesis. And so they can they can grow new organs in response to the environment. So I think part of it is because of that, right? And so you do see this in the plant kingdom in other interactions. So the good example is the beneficial symbioses. These also make new organs for the kind of nitrogen fixation and that kind of thing. So,
nodules.
Yeah.
So you work with cyst nematodes. What systems is this in? Is this potato cyst nematode? Are there several? What systems are you working in?
Yeah, there are several. And so I think, cyst nematodes can encompass a lot of different crops and a lot of different plants. We started out doing potato cyst nematode mostly. And that's because the potato cyst nematode is the number one cyst nematode in the UK from a kind of agricultural perspective. We grow a lot of potatoes, and something like 50% of potato growing fields are infected with this nematode, and so it's a serious issue. And the farmers are really quite rightly so panicking because a lot of the control measures that they used to be able to use have been taken away from them. So a lot of the chemistry they could use is gone. So there's a huge drive to do that. And so we did a lot of work in potato cyst nematode. A couple of years ago, we started thinking about, trying to build a system where we could answer more ambitious questions. Because there's some really, working on potato is not easy. Working on potato nematodes is not easy. There's lots of things to do with both of their biology that makes them complicated. And so we looked for a model system that was closely related, still a parasite, and that we could really explore more deeply and port over information. And so we chose what's called the beet cyst nematode. So Heterodorachectii, which is about as closely related as you can get, but that nematode can infect the model plant, Arabidopsis thaliana, and so that just makes, then you have all of the power of the model system at your disposal. And so we kind of, the lab is kind of split on both of these, and I would say actually more and more we're doing this model system, but always with the view that, the reason we're doing it is because we want to understand the nature of this system so we can port that over to the systems that really, really matter.
You mentioned, and I've heard about this in previous podcasts, one that I did with Matt Back at Harper Adams. I was kind of curious as far as just the drivers of nematode research in general. I think that the UK and the European Union is really interesting, an interesting contrast to the US in that, like you said, a lot of chemistries are being taken away as far as resources for management. So I was just kind of curious, I'm not super familiar with agriculture in the UK and that kind of thing, but is that the main driver as far as kind of pushing the boundaries here, or what would you say that is?
Yeah, I think so. I mean, look, I don't know a lot about agriculture either.
Sure.
Right?
Okay.
So I don't know. But it definitely feels good to work on a problem that is important.
Yeah.
Okay. And it definitely helps when you're trying to convince someone to fund your research that the problem is a big problem, right? Yes. And so it definitely helps. And so, there's this kind of two prongs to this. You actually want to do good. And, if you can convince somebody that you actually want to do good, then that really helps. So I think that's kind of one of I would say that's one of the main drivers. Although, let's not forget, there's really interesting biology underlying these, right? And so I think there is a case for just looking at them from a fundamental biology point of view.
Absolutely.
But I think it's not the dominant driver. The dominant driver is the problem.
Sure. So I did a little bit of research on your lab as far as some of the, what I'll say to me are innovative methods of data collection that your lab is doing. So I know I saw you guys were doing some 3D printing projects. You were using some machine learning and some artificial intelligence to kind of analyze the data gathered from these things. Can you tell us about the project that you're using before we get, because I want to get in and really kind of nerd out on what you guys did with each of those aspects. But can you give like an overview of the projects that you guys are using this equipment for?
Yeah, sure. So maybe I'll just start with a quick overview of the kind of things we do in the lab.
Sure,
yeah. So we, fundamentally, and this is going to sound abstract after what I've just said, but it is true. What we work on is the communication between plants and animals. Okay. And this is a molecular communication. So we work on the kind of molecules or proteins that are involved in that dialogue. And that kind of that's the kind of core fundamental question that, kind of underpins all of the, ideas that we've talked about, that we want to protect the crop, but you do that by breaking the communication.
Yeah.
Right. And so that's the kind of underpinning thread. And so the lab has kind of these two main sides. One is understanding what are the nematode genes that contribute to this communication. Right. And then the other side is what are the plant genes that contribute to this communication. And so this project was really on that plant side. So the lab does both. And this is kind of, something like half the lab maybe works on this kind of plant side. And the kind of, one of the ways in which we're limited on the plant side is simply this measurement problem. So, oftentimes, you want to do these experiments where you're measuring the phenotype of something in some kind of large scale. And it's simply your ability to measure is what limits you. And so I don't know I can't remember who did this. I think it might be, I should have done my prep beforehand. I should have worked this out. But there's this wonderful, these quotes that you don't know the quote, but you know the sentiment of the quote.
Yeah, I do it all the time.
Good. Okay. I got one of those. And the idea is that progress in science, people think progress in science is made by, the lone academic sitting in an office, thinks up a brilliant idea, and then the world changed. right?
Yep.
That is not how science is made. What happens is somebody makes a technological advance, okay? And you can measure something new, and only then is progress made. And it's something like 70% of the kind of progress in science is determined by technological advance first, and then conceptual advance. And people don't appreciate that. And if you think about it, it's kind of obvious, because if you can't measure it, you cannot hope to understand it.
Yeah.
And so this project was all about how can we measure better the interaction.
And just in general, again, you're looking at Arabidopsis plants with Heterodorachactii, the Beatrice nematode, as your kind of model system that you're using this project. So you're looking to advance data collection, right? So what was the inception of this? Because I'm assuming it started off as an initial plan and then it evolved as you went.
That's always the case. Yeah. I think this is going to resonate with some people and not at all with other people. Yeah. So in genetics, there's two ways you can study gene function. There's reverse genetics and forward genetics. Okay. And so reverse genetics is you have some process and you want to find a gene involved in that process, and basically you make an educated guess. So you say, I think this gene is involved in that process, and then you test whether it is. Yeah. Okay. And that is rough. Okay.
Yeah.
It's really hard going.
Yeah.
I've done a lot of that. And it is really, really hard, because you have lots of genes and none of them have functions, right? And phenotypes, and you have no idea what's going on, right? And I've done that at the beginning of my career, I was doing a lot of that. And that's really hard going. And so I always wanted to do the opposite, which is forward genetics. Okay. And so this is where it's the other way around, and you screen lots of stuff and you look for the things that have a phenotype, so they look different. Yeah. And then you find out what gene contributes to that. And that's also rough. Okay. Yeah. But what's nice about
scale, right?
Well, also, but even just to find the gene can be hard. But at least you have a phenotype. Okay. So at least you have, something you're measuring, they look different or whatever. You've got something, right? Yeah. And in order to do this kind of forward genetics, you need that scale, basically. And so that was the kind of, I'd always had this in the back of my mind. I wanted to do genetic screens because, again, this may or may not resonate, but, genetic screens for me are one of the most elegant ways you can do science. They're just beautiful. When you see a really beautiful genetic screen and it's just really clever, and it's one of those I wish I did that kind of experiment. Yeah. It's one of those. Yeah. So I always wanted to do one. And in this case, we thought, how can we do it? It's just sort of not possible on the nematode side, and especially not when we're doing the potato cyst nematode. But when we had this model system, genetic screens are done in Arabidopsis all the time. Yeah. And so we're thinking, okay, now we can actually do this, right? What would we need? And what we need is scale. Mm-hmm. So this is where we start thinking about, how do we build scale? What do we do to, get scale?
So what does this look like? In your current research, just a little bit of, getting ahead of myself a little bit before we dive deep into what you did. But it sounds like you have a greenhouse scenario with a bunch of Arabidopsis plants, and you've created I'll let you explain this further, but this is my summation of what I heard and researched. Was you've got a clear growing media for the Arabidopsis plants so that you can see what's going on in the root system with the nematodes, and then you've got a system to more or less photograph it, analyze those photographs, and then give you a bunch of data. Is that?
Yeah, that's
a pretty good summary.
That's perfect. Yeah, exactly. So if you break it down, these are root parasitic organisms. Okay, yeah. And so first thing is you can't see them, right? If they're underground, that's problem number one. You got to solve that. That problem has been solved. We didn't solve that, and so you can grow things in transparent medium and agar, and this is sort of a relatively well established system. Works really well. And in fact, in the model system, it's really easy to do this, so you can do that. And so number one, see the roots, see the worms. Okay. Then obviously you've got to have a way to measure them. Okay. And the easiest way, the most intuitive way is obviously you take a picture and you measure from the picture, and you can do that later. Okay. So what it used to be, and it still is in some small cases, is, humans counting, so looking at these, we talked about this earlier, under the microscope with your clicker, clicking away. Yeah, exactly. It's bringing back memories.
Yeah. Or flashbacks of horror. I don't know.
Exactly. So that kind of thing, right? And so now we do still do that, and in fact, there's some things we have to do that. But that's the way that is. And so you've got to think about how do you do this in a different way. And obviously, with AI, we did this a couple of years ago. AI was just sort of coming along. And so it looks like an obvious way to be able to do this, right? If you can get a picture, you can measure from the picture. And so that's fundamentally the idea is we thought we would build off this established system where we know we can grow them really reproducibly, relatively small form factor. So, something like 5 centimeter Petri dish. So it's quite small.
Oh, wow.
Yeah. So like this big, right?
Yeah.
So, yeah. And in that you can grow one plant. You know the genetics of that plant, and then you can infect with nematodes, and it's a totally sealed system. It will live for, 60, 90 days, no problem. Nematodes will go through multiple, you don't have to water it, you don't have to open it. It's totally sealed and sterile. And the nematode will go through multiple rounds, so they'll infect, and then you'll get a secondary infection. So you can track everything and see it beautifully. And so then, what we did was build these machines that allowed us to kind of, do this really high throughput imaging, and then the AI layered on top of that to do the measurement from the images.
So just in general, do you have like a growth chamber where these things are growing? Is it, because you mentioned sealed systems, because obviously you don't want things falling into these, the air is littered with all kinds of things that would love to grow in that media. What does one of these experiments look like?
It's way bigger than you think. Way bigger. So in the kind of, the big experiment we're talking about, it was 10,000 of these Petri dishes. Yeah, but that sounds like a big number, but until you've seen 10,000 Petri dishes, you have no idea how big is 10,000 Petri dishes.
Yeah.
Okay. So, and it was such a logistical challenge, even for basic things, like you have to autoclave an ungodly amount of media in order to, you know what I mean? You have to dominate these flow chambers for just months. It's like months of work just to prepare all the media, and then you got to store it somewhere in the cold, and so it's literally rooms. It's a huge amount of stuff. And so when you lay it out, I'll try and give perspective. So when you lay it out, 10,000 Petri dishes, as tightly touching one another as possible let's get this right. So we had 4 benches that were 11 meters by 3 meters to carry those 10,000. So it's like a massive room. So one of this room is probably a quarter. Yeah. If you lay them out, it's something like that. So it is an unbelievable amount of Petri dishes. But it's one of these nice round numbers, so it feels kind of good, right?
Oh my gosh, I have so many questions, but so you've got this massive amount of Petri dishes laying out with Arabidopsis plants growing in them, inoculated with cyst nematodes. There's still manpower involved. It's funny because when I was coming into this, I'm thinking to myself, man, okay, he's using these tools to kind of eliminate a lot of the manpower needed to do this massive experiment. But what I'm hearing is there's still how do you set this up? Do you just have an army of students for a week?
No.
Laying out? It's
this was a full lab project. So firstly, I should say, so we're building this hardware. We're sort of designing and building the machines and 3D printing them ourselves, right? And maybe we'll talk about that later. But we're doing this live while we're pouring the plates. Okay, so we're in this. Okay, so we are making the plates. We've invested in that. This guy's pouring them, and so if you don't get the machine done on time, you have a massive so there's a deadline, right? And so we're developing this in kind of parallel, and it's kind of an all lab type project, right? So everybody got
all
involved.
Hands on deck.
No, literally. And so yes, the machines we built are kind of semi-automated because there are things machines are good at and there are things humans are good at.
Yeah.
Okay. Yeah. And so there is no point making machines for things humans are good at when you have the humans around. That doesn't make sense. And so you want to work together, right? So, and we're not engineers. We are, synthetic biologists, engineers, geneticists, whatever. And so we're not, we don't, so there's some challenges that are too much, right? Yeah. And the final kind of piece of the puzzle is, we want other people to be able to use the technology. And so the less complicated is the better, basically, right? And so, you don't want this totally complicated solution. And so our solution does involve some human labor, obviously. Yeah. And that human labor is quite extensive. So, you're imagining this greenhouse with these 10,000 things. It's 40 meters by whatever, 15, 12, 15. And so, there's a huge area. And then in that greenhouse, we put these imaging machines, these kind of machines that we built. But then someone's got to go put the plates into the machines and then put the plates, these petri dishes back in the same place they were in the greenhouse. And so that is a lot of work. And so it was 4 people, 3 hours at least, 3 to 4 hours per day to pick up 10,000 petri dishes, run them through the machines, get them imaged, and put them back. And so everybody in the lab helped out. I mean, everybody, right? Yeah. We had this and it was every day. It wasn't, some days weren't just like every weekday. They were every day in the beginning.
Yeah.
And so we had this Google Doc and people would sign up. And obviously there's some people that were sort of peripheral and so they would sign up less often. That's totally understandable. Yeah. And there are some people that were really core to this and they would sign up often. And, we had Thomas Baum, one of your faculty. He was visiting. He signed up, So it's amazing, right? And so in the talk I'll give later on this afternoon, there's a picture of him doing it.
Oh, amazing!
In the greenhouse. Yeah, it's so good. But yeah, so everybody helped out, and I think I think there was this. It was just like super exciting time, right? And so everyone was really just into this, and I think there was this. It wasn't hard to convince people to sign up by themselves, to do this. I would sign up obviously as well and do as much as I can, and so it was kind of easy to convince people to do it, but it was not easy work. So it was like back-breaking work, and like you could ask Thomas about this, but it was like hot in there, and like it's pretty heavy going and stuff, so it was pretty rough. And so we did that. We imaged all 10,000 plants, 45 times over 90 days. So it's basically on average every other day. Yeah. Now it was harder than that because in the beginning we actually did it every day. Yeah. And there's kind of a biological reason for that. And then in the middle we also did every day. Sure. And then we spaced it out, and you'll see a graph in the talk later.
I can't wait to see pictures of this.
Well, I'll show you this, and it's so funny because we've got even just simply a graph of the sort of number of plants we imaged per day over time, and you can see these things that I described, this when we do higher frequency and lower frequency, but also you can actually see the fatigue of the lab. So, like at the top of the curve, they start getting spaced out. You know what I mean? Yeah, it's getting hot. It's July at this point. And so, it's much harder to convince people to do it. And so at some point we got to end, you know what I mean? And also, they've done this like 45 times now. So it's getting heavy. So.
Did you like take some before and after pictures of your lab, like the people and how they looked?
Oh gosh, see if it's aged. No, I didn't. I should have done that. Yeah.
So so it's funny because I don't think I had a, my vision of what this looked like, was. It's funny. I was just doing a podcast the other day with somebody, and I've been referencing this way too much, so I'm sorry. But I was referencing the Jetsons, and like this future. I don't know if it's a cartoon, an old cartoon where somebody was trying to predict the future of humanity, and they were flying in cars, and they had robots doing all the work for, basically like. Not where and I think that was like where they thought we would be by like the year 2000,
yeah,
like
exactly,
at the time, right? Yeah. And anyway, like I think that my my Jetsons vision of this was that you literally had robots scanning over these things back and forth over rows of, but no. So what did the machine look like?
Yeah, I wish I had a.
I'll try to grab some pictures from you at some point to throw them.
I mean, look, it looks like we designed it, right? Okay, in every way.
Looks like non-engineers.
Totally non-standard. Yeah, sure. Yeah, exactly. So I mean, it's a bespoke solution for a bespoke problem. So it doesn't look super shiny and slick and everything, but it does the job. So you can imagine, there's it's this. So let's think about this. So it's something like, 50 centimeters wide, something like 40 centimeters high. Yeah. It's got these 2 kind of stacks on either side. One is the kind of input stack and one is the output stack. And you stack up all these petri dishes on one side and the machine will pick one at a time, move it under the camera. It gets illuminated in a specific way and then it gets returned to the other stack. And so it's not like set it and forget it, like you're describing, but you can set it and forget it for a little bit, so you can like load up 20 plates and then you load the next machine while that one's running. And so functionally how this worked is we set these up on a bench like this. And so there would be people doing the loading and people doing the unloading. And so, you kind of, that's the logical way to do it. And so we had these 6 machines, I think, in parallel. And we would then have I think it was 2 loaders, 2 unloaders or whatever. And you would kind of, process them through like that. And that seemed to be, we kind of, obviously we were finding our way, but we kind of settled on that being the way to do it for these. But yeah, I mean, people do have these kind of set it and forget it systems. That's not our solution. Yeah.
Well, so within this this machine, I'm assuming there's a camera. What else is involved in there? You said there's a particular light or angle of light.
Yeah, also, yeah, very much so. So Raspberry Pi. So some of your listeners might be familiar with this. So this is a small computer called a Raspberry Pi, and it's used for this kind of. People use it for all kinds of things. I use it for home automation. People do it for that kind of all this kind of small engineering projects, and you can code on it really easily in Python. So you can do these simple kind of loops, simple if type statements and stuff like that. And so that's the kind of logic that we could use to the brain of the machine. And then the body is, simple, low power motors, stepper motor type things, lights that could illuminate in a very specific way. So it turns out that the angle of the incidence of the light on the petri dish really, really matters. Otherwise you can't see the nematodes well. And we had to kind of, we had to dial in all this stuff and, a particular camera. So I think it was can I remember, 21 megapixel camera maybe, with this telephoto lens so that it's, as parallel light as possible. Yeah. So you get, less kind of fisheye and less warping on the edges and that, you know what I mean?
Yeah.
That kind of thing we had to dial in. And then the motors drive these wheels that we designed to kind of select one dish. But of course you need to know where the wheels are. And so these are indexed by a Hall effect sensor. So this can sense magnetic fields, and so you put a magnet on each one of the teeth of the dish, and then you know exactly where you are, so you can kind of index, right? And so maybe it sounds complicated because I'm describing it badly.
No.
But it was fundamentally, it's super, super basic, okay? Super basic. You just got to pick one dish, move it to the middle of the machine, stop, illuminate, take a picture, move it to the other side of the machine, repeat.
No, I think you explained it quite well. I'm tracking. The face I'm making is more like I'm curious. This is a complete sidetrack, so apologies, listeners. But I'm just curious. You mentioned, you're not engineers. This sounds more complicated than something that I know I could come up with. How was there an inspiration for this? Or is this something that you had a vision and you kind of came up with the components to make this machine? Or I don't know. Yeah, what was that like?
Yeah, I mean, it evolved over time. So look, I'm reasonably handy, okay? Yeah. And there was a couple of guys that had done some electronics. So one of the guys in the lab, Olaf Kranse, one of the principal authors on this, he's really good at, low power electronics and that kind of thing. He had been doing that in his free time.
Yeah.
You know what I mean?
Yeah.
And then there was a
lucky hobby for you in the lab.
Yeah, but everyone came together, right? So he did that. I had done some 3D printing, already in my free time. And, I think that we had the IT technician at the time, Igor, I think his name was. He was a photographer in his free time, and so he knew about f-stops and that kind of thing that we knew nothing about.
Yeah.
And so we all kind of just came together and just sort of got through it. And you can learn a lot on YouTube.
Yeah. Yes.
And so there's these kind of engineering principles videos on YouTube that show you different mechanisms that would perform different functions and stuff like that. And so we can kind of cobble it together from that kind of stuff. But look, you would be surprised how I bet you could do it. Yeah, it's not that bad.
It'd be a lot of time on YouTube, probably.
Yeah, but it is. Yeah, but it was a super exciting time. It was the best time in the lab actually for the last 5 years, because we were just, we were really. I was talking about this earlier with somebody. Discovery feels good anyway, right? When you find out something new that nobody else knows, that feels great. Yeah. But when you built the thing to then do the discovery, it feels so much better. You really feel like you're pushing something forward, because this tool doesn't even exist, and then you use it to find a thing. And maybe we'll talk about it later, but this is the kind of thing where now it's that approach has infected the lab. That the approach is we almost choose problems by those that so. Is there a machine that can measure this problem? No. Good.
Yeah.
Okay.
Yeah.
Can we build that machine? Yes. Really good. Blah, 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 blah. Yeah. And so I really like the idea that you have this problem. We cannot answer this problem. It comes back to what I was saying about technology, right? Yeah. So we can't measure it. So let's build a thing to measure it, and then we solve the problem. And it's kind of this core ethos is a bit of a strong word, but you know what I mean? It's we really, it's really nice way to do science.
I like what you said there about, I don't remember the exact word that you used, maybe infectious for the lab. That's got to be incredibly fun to, I don't know. It probably just boosts. I mean, you mentioned like how the lab members got tired over time, but man.
Well, obviously, yeah.
Well, yeah, but at the same time, there's still this underlying thing of look at this thing that we made and it's working.
Yeah, you could feel that they could tell this was a big deal.
Yeah.
Yeah, you could feel it. It was cool. It was the best time. Yeah, and also, for me, I don't do a lot of hands-on science anymore.
Yeah.
Right. I do mostly,
sure.
type on a keyboard. And this, I actually did some of the, the engineering bits and stuff like that. And so it was really nice for me to kind of, do that again. And it just was really good. Yeah, it's a really good time.
So you have all the petri dishes, you have the machine that you're loading them through. I'm at this point, like this part is quite, I mean, I don't know if I can even say that it's the most interesting part to me, but it's something that I, like I said, I spent a lot of time counting nematodes with a clicker. So now this is the part that's really interesting to me is now you're taking these photos and you have to train a model to identify the nematodes from the photos. Is that the next step, basically?
So firstly, I should say that even if you don't train the model, it's still good. Okay, because we have digitized the phenotype at that time. And so, if you had 10, even if you had 1,000 plants, you can't even count them in a day. And obviously it took you a day. So, you're just not going to do it, right? And so even just by taking an image, you're part of the way to solving the problem because you can count in the future. So it is part of it. It's a good step. And, but obviously it's better if you cannot count them manually, right?
Yes.
And so that's where the AI comes in. And so, so this, we tried to do this alone. So we, obviously, as you can tell, it's kind of, we're kind of people that like to try and do stuff if we can. That was a bit too hard. So we had to collaborate with AI, proper AI expertise to be able to do this, because it's just really, really tough problem. And maybe as I'll describe, it's a particularly tough problem. And so in this case, what we did was, we actually trained 2 neural nets to do this. So depending on how much people know about neural nets, good analogy would be, when you point your phone camera at somebody, then oftentimes there's a box around the face of the human. And so that's called an object detection, right? And so it's just saying there is a human face in the screen, object detection, detected the object. And so we trained an AI that does that, but for nematodes, right? And that's one part of it. And that does your simple clicker counting. Yeah. Right. That is the count.
There's a nematode here.
There's a nematode here. Easy peasy. Yeah. The next one was harder, but better. And that's what's called segmentation. And so this doesn't say that there is a nematode here. It says, draw a line around that nematode. So distinguish it from everything else in that little box. Gotcha. And that's super powerful because then you get lots of information about the nematode itself, not just that it is there, but, anything that you can measure from an image, you get. So you get how big is the nematode. That matters because, the amount of nutrition they get from the plant determines how big they get. And so the bigger they've drained more. You get the shape. And so you can tell whether they've laid eggs or whatever. You get the color. So you can tell whether they've, matured and completed their life cycle and all this kind of stuff. So, inherent in doing a segmentation, super powerful. And so we also trained a model to do segmentation. Now that was way harder than object detection.
I can, yeah,
I didn't, I didn't have, a preconception about which one would be harder, but it just was harder. Yeah. Okay. And it turned out that actually, we used a foundational model that somebody else had trained called Segment Anything Model, SAM. Okay. So this is from Meta, so from Facebook. Okay. And they had trained what is called as foundation models, which just understands the relationship between objects and pictures. And so it's been trained on just lots of photos, and people have just segmented, this is one object, this is another object in the photo, here's another object. And so this model has built this kind of understanding about what objects look like in images. Yeah. And that already, out of the box, even though it's never seen a nematode, just knows this is an object. I know, shocking, right? In fact, it's utterly depressing that the model the one that we trained was worse in the beginning than Sam. But then you can adjust the weights, right? And so we can kind of retrain it. And so we built what we call NEMA-SAM, which is sort of building on this and sort of, taking that basic foundational model, tweaking it to be really, really good at the nematode detection and then that. And so what we got is these 2 neural nets, one we kind of did from scratch for object detection and one that we kind of built on existing foundational model, and you combine the two. Yeah. And so one model says, here's the nematode, and the other model draws the line around that nematode. And then next one. And in that way, you build up this kind of quantification of the parasites in each picture.
Is it able to detect the female nematode like in vermiform state as well as once it's
Yeah, so there's a limit to the detection.
Okay.
Right? So if you go backwards, if you imagine from like a large female, which is sort of quite macroscopic, you can easily see with your eyes several millimeters across, right?
Yep.
So quite big. The kind of vermiform stage is like, 0.1 millimeters wide or something like that, right? And like less than a millimeter long. You can't, unless there's a million in a tube, you can't really see them, right?
Got it.
And so there's a limit to the resolution of the camera, obviously. Some of these worms are going to be, in the beginning, they're inside the root.
Yes.
And the whole animal is the width of one root cell. So you are not going to see that.
Okay.
But at some point, they grow to the stage and you can see it. And for us, it's about 7 to 10 days, something like that. And they're sort of largely in this kind of, I guess we would call fusiform, sausage shape. And then they become, much more kind of, they kind of swell up like a tick kind of thing. And so we track them as early as we can. And then we kind of, it's perhaps a bit too in depth, but you can infer. So we know the size they all were because all of the nematodes are born the same size.
Yes.
And so when you're building your kind of growth curve of the nematode, we actually know what size they started at because they all start the same size. And so then we can infer. And so even without incomplete data, you can interpolate and get your perfect.
Got it.
Yeah.
That's so cool.
Yeah. I should say that the final kind of piece of the puzzle, which is worth mentioning, is that because our way to measure is we take an image. And so there's no reason you can't image the same plant tomorrow, right? And provided you know where every nematode is, you can line up today's nematode with tomorrow's nematode. Yeah. And so what you end up doing then is tracking each individual parasite over time. And that's how you build these growth curves. And so what we've got is this sort of like hundreds of thousands of images, but actually each one of these is part of a series of images, right? So this kind of like a stop motion movie, if you imagine. And then we track the individual parasites, all of them per host on 10,000 stop motion movies. And so we have this incredibly rich data set of these growth curves of, my goodness, how many? I think it's hundreds of thousands of worms on, tens of thousands of plants,
You must have quite the cloud storage vault going for this project.
Yeah, it was heavy. I'm going to tell you a fun stat. So I remember when we were doing the imaging, we had to swap the hard drives on the imaging machines every day. So it was, one machine was 300 gig of data a day.
Yeah.
And so we just couldn't actually transfer it off quick enough. So we would just swap them over. So we just swap the hard drives over. And so, we're like utterly paranoid about, so we have a lot of copies of this.
Yeah.
They're in physically different buildings. Yeah. We're not doing this again. Not anytime soon anyway. Yeah.
So you've got this data in these images. What's the next step as far as using it?
Yeah, sure. So there's a big bifurcation, right? And so what can you do with the information? And so what we've got is big numbers, okay? And to give the big numbers in perspective, this one experiment, we measured more plant parasitic nematodes than have been measured in all previous publications combined. Okay, so it's really big numbers.
Yeah.
And in some ways, in measurements that are not just like counting, but like size and shape and color and roundness, etc. So we've got this incredibly rich data set. And so what can you do with that? And you can do two things. You can do fundamental discovery, and you can do impact. Let's put it that way.
Sure. Yeah.
So fundamental discovery. If you're looking at millions of worms, you can make assertions or discoveries about the kind of fundamental nature of those systems because you have seen it in so many examples, right?
Yeah.
And so there's a whole bunch we have done, but I'll give maybe one example. And so one of the questions is, if two nematodes are infecting the same part, two parasites infecting the same part of the host, do those parasites help each other or do they hurt each other? Okay.
Yeah.
And there's actually good biological, basis for both. You can rationalize both in your head. And because they kind of, the feeding sites that they form, these organs that they form can actually fuse. And so they actually share feeding sites, for example. And so you can imagine that they might be competing for resources, or you can imagine that they might be helping each other out. It could be that twice as many nematode, genes making this organ makes a twice as good organ, or more than twice as good. It could have some kind of more than additive effect. So you could rationalize it both ways. And so we don't know the answer, but we have million nematodes. So we can go and find out. And so we can basically find all the examples of nematodes on all of the different plants. And we can say, how close is your nearest neighbor? So how much space do you have until you touch your nearest neighbor? Basically get this distance. And then you can plot that against how big do you get? So how much nutrition do you get as an individual parasite from the plant? And what we find then is actually we have this really, really like strong local negative effect of individual parasites on one another. So they sort of have this detrimental effect on one another. And so, our original idea was that they actually do compete for resources, but we've actually delved deeper into this, and they don't even need to be sharing in order to have a detrimental impact on one another. So it's just their proximity. So we don't have any idea, or we don't have any good idea about why this might be, but they negatively affect one another. That's new fundamental understanding about the system, which excites me at least. And we can only answer that because we have such big numbers. So it's the kind of thing that you need such huge numbers to kind of, filter out the noise and really see the true, what is the signal. So that's one type of thing we do, fundamental discovery. The other type of thing we do is obviously trying to be useful. And so this gets back to the reason we did it, which was a genetic screen. So what we wanted to find was the plant genes that contribute to these nematode traits. And so, these 10,000 plants wasn't just random 10,000 plants. Obviously, this was 550 different genotypes, so different accessions, and each one replicated 20 times. And we already know, the sequence of the genome of all 550.
Yep.
Okay. So what you can do is you can kind of correlate differences in their genome of the plant with differences in how those nematodes look with what we measured from the AI, right? So either how many there are, how big they are, how round they are, et cetera, et cetera. And so then you can make a correlation between what are the genes in the plant that, associate with these nematode traits, nematode phenotypes. Yeah. And so then you can, there's a whole bunch of mechanisms you can do then to narrow down what are those genes and really find the kind of, the causal gene. And so we've done this for a couple of examples where we know, okay, when we take this gene from this plant, it does confer resistance or susceptibility or whatever to the nematode. And so that's trying to be useful. I mean, there is way more than we could ever do as one lab. Yeah. And so, this is the kind of information we'll give out and say, these are all the loci in the plant that we think contribute. Feel free to go and have a go kind of thing.
It reminds me a little bit. I did some podcasts. My perception of time is warped some time ago.
In the past. Yeah.
About, doing phylogenetic analyses of a lot of different fungal pathogens, right? And I think somebody told me that they had enough work that they could employ 10,000 graduate students for the next 20 years or something.
Yeah, and that's a lot.
Well, yeah. I'm paraphrasing.
Yeah,
it was some crazy number.
Yeah, I think that is the world we live in. Is we have way more data than we know what to do with. And I think that's across the board. It's not just us. I think that's generally true.
It's amazing to me the advancements that we've made and how far we still have to go, right? And it's very inspiring to me.
That's a really interesting philosophical question about whether science gets bigger or smaller. So, obviously it's getting bigger, right? But whether there's an end, it's not obvious to me that there is or isn't an end. I generally come down on the side that I think there probably is, but it's not necessarily the case, right? And so, definitely right now we're in the phase where every discovery you make increases the number of questions that you have, not decreases it. Yes. And so definitely we're in this expansion phase. And, I think, coming back to AI, that might be one of the ways we help solve this. So there are, there's a lot of work going on with kind of agentic research AIs. I don't know a lot about that, so don't ask me too many questions. But there's so much scope for what you need is more intelligence to look at the problem, and that might not need to be biological intelligence. It might not need to be people for some of those problems, right?
Yeah.
No reason that it should be, basically.
So, I think at this point that's kind of a good transition into what. That's kind of a good picture of what your lab is doing now. What's the next steps? Where do you plan to go with this stuff next? Just collect more and more data I don't know.
Yeah, I think so. I think so. One of the amazing things about this data set is that we still have it. So we can go back.
Yeah.
So we can just have a new idea. Yeah. And so you just build a new AI that measures something else, and you go back into it and you can measure that. And then, because again, everything's already sequenced and we have all the images. So you can just immediately rerun the pipeline, right? So there is a lot of work to do there that we could do, and we are doing. So one thing we're thinking about a lot now is not just measuring nematode phenotypes, but measuring root phenotypes. So, from an agricultural point of view, sometimes when nematodes infect plants, you can have a really negative effect on the plant. So even though you don't get many nematodes back, you don't get much plant back either, right? And so that's not good. You don't want that. And this phenomenon is called intolerance. Yep. And so there are some resistant varieties that, yes, you don't get many nematodes, but you don't get anything else either. Right. And so that's not good, right? And so we can start to tease apart that because if we measure the root from the images, the amount of root that they produce, then we can get a proxy for how well they're growing under infection stress. And so, but of course, what we need for that is an AI that measures roots. And so, that's the kind of thing we can work on. So we can go back into it. There is a lot of, the genetics that we can do and find genes involved. We'll do some of that. We won't do most of it. I think that'll be other people's work. And then there's the kind of, what else do you do after? So I think there's probably two things that are worth saying. One is we are building a new machine.
Yeah. Okay.
Okay. So, and this new machine solves all of the problems of the old machine. So the fundamental idea at the beginning was to move the petri dishes. Turns out that's a terrible idea. So there's all of the kind of like difficult parts of the engineering were because we were moving the dishes. So it's better not to. Oh, okay. And so actually we can basically now the unit that you move before was one dish. Yep. And so actually we're going to scale that. So we will build a kind of a cartridge, right? A shelf. And so we move the shelf and we'll move that onto a machine that has 0 moving parts. So instead of like having one camera that moves, we just have 50 cameras. Like it doesn't even cost, they don't even cost anything, so they're basically free. And so you can just have this like crystal of cameras is what we think about, and just illuminate everything, flash it, and then the shelf goes and you get the next one. And so the number of actions that the individuals have to do is lower. Yeah. The moving is lower, so these things last forever. You know what I mean? So it should be way better. And if you're not having moving parts, a lot of the kind of, there are a lot of problems that emerge when you image the same plant in 2 different days, but between that you moved it, right? Because you put it back, and so you might not image it in the same orientation or exactly in the same way.
Yeah.
And so that's a computational problem we had to solve, and I don't want to have to solve that.
Yeah.
You know what I mean? So there's a lot of things that it would be good if we didn't have to do that. There's a lot of other stuff, but so we're building a new machine. Yeah. And so the implication, of course, of building new machines is we will be doing more screening, and so we do have plans for that. The other one that I really, really want to do, but we don't have a solid plan for it right now, is there is another nematode that can infect the same plant, but it evolved the ability to make the plant make a new organ independently. Okay, so this is the root knot nematodes, right? And so they can make these new pseudo organs as well, but they do a different kind of pseudo organ, has the same function, but they do it differently and they evolved independently. And so then you can imagine when you've got 2 such big data sets on the same set of plant genotypes, you can start to understand what are the shared features about this. What are the shared features if you've got to interact with the plant in this way, and you communicate, it comes back to this communication between kingdoms, and what's the unique features. And I think that will be from like an evolutionary point of view about, communication between animals and plants, molecular, what is the, common and unique features. That would be super cool. So I would love to do that. That would be cool. I suspect we'll do that on the new machine, but I would love to do that. So those are the kind of things, so, going a bit deeper into the genes, going a bit bigger, different species, getting this evolutionary view, I think would be really cool.
It's funny, from a hardware standpoint, I was just thinking, I was thinking back to the days when you'd go and you'd buy a flash drive, and like a 500 megabyte flash drive was, there was a point in time where that was not a cheap item to buy, and now you're talking like
it's true. Yeah.
Tiny cameras, Raspberry Pis, just it's like a dime a dozen.
It's
kinda
shockingly cheap. I mean, so let's put it in perspective. One of the machines that we built originally is something like £200.
Yeah.
I mean that in a kind of a science, the cost of science is quite high generally, and that is nothing. If you most of the cost of science is people. Yeah. Okay, and so that's almost invariably the case. And so this next to that is just dirt cheap, super dirt cheap. And so I think it is super exciting, a time that we can maybe, was it Jetson?
The Jetsons.
Yeah. It is this kind of futuristic view that you have this kind of replicator in your house or whatever, and you can just you can print these things, and it really is that kind of vision that you can just design what you want, make it, and then and do the science. And I think that's like super exciting.
Well, I think with that, Sebastian, is there anything that we missed with regards to this research and kind of what you guys are doing before I get to wrap ups?
Yeah, I think
anything else that you want to talk about? I mean,
no,
fair game.
No, we're good. I mean, look, the lab does lots of work, right? And so this is only a small part of the lab, and I don't want to, make it seem like there's other things in the lab that are not as important. We do lots of cool stuff that's kind of relevant. And I should kind of go back to the beginning and say, this is really one part, and it's probably not even the main part these days. The other parts we're thinking about are more just this communication. Maybe this is a nice one to end on. So when you think about a communication between plants and animals, which sounds esoteric, but happens all the time, what is the information that transfers between them? In what way is it encoded? So what is so is it a protein? Is it a lipid? Is it a sensation? So is it a physical touch and things like that? And we're starting to think about that. And I think this is quite an exciting direction for us, because it really gets at that core underlying communication between the kingdoms. So
it's a huge stepping stone, right? For, like you say, like you want other people, other labs to kind of use this as that stepping stone and see what all the avenues people can go with it are. That's exciting.
Times.
So that's perfect transition into first wrap up question I have for you. So as the field of nematology advances technologically, how do you think that this is going to change or affect nematode management?
Tough question. Yeah. So I think, the idea is that you have this new capability to screen plants for nematode resistance. That is how you get a resistant plant. Okay. So if you're doing breeding programs, that's what you have to do. You have to screen, even greater diversity of plants, actually. 550 is a kind of a small number when you look at big breeding programs. They'll screen more. And so anything you can do to accelerate this process is good. And so, I should say, look, really clearly, we did not build this for breeding companies. We built it for our lab. Okay. And so it is highly unlikely that the solution that was optimal for our lab is the solution that is optimal for their labs. I suspect it's not, because they're going to have different processes, and they're not going to be doing this in petri dishes, for example. But that's the kind of idea that if you can automate these kind of processes, then you can accelerate the breeding. So you just need a better way and a different way to measure nematode infection. Just to kind of linger on that last point, historically resistance has meant the number of nematodes, right? So that's typically what you would measure in a breeding program, would be like how many nematodes. Yeah. But actually, what you really care about is how many eggs are left in the soil at the end of the season. Okay. When you harvest the plant, how many eggs are there that are going to infect the next one? And there's lots of things that go into that, and we don't often measure them, but we can measure them in this type of approach. So for example, what you don't want, 10 tiny nematodes is obviously a lot different to 10 massive nematodes, right? And so massive nematodes are going to give more eggs. And so you really want to be able to integrate these two things. And so that's a simple example, but there are others. So, how long does a nematode take to develop, right? And so one of my favorite ideas that I don't think is useful, but I really like it, is that what if you could, so how long the nematode takes to develop is controlled by plant genes. There is variation. And so therefore you could have plants that allow the nematode to infect, but it develops so slowly that by the time you harvest the plant, it's never finished. It doesn't finish. And so you might have nematode infection this round, but there's no more in the soil for the next round, right? And so I really like that idea. There's something about that really just feels, I don't think it's going to be useful, but it feels good. And do you see what I mean? So we need I think a more nuanced approach to what does it mean to be resistant, and what are the genes that we really want in those resistant plants in the breeding. And so I think that's the kind of principal, benefit we would have on this, nematode management.
It's interesting. Like, if you ask, I think, any plant pathologist, like what's the most important management of any pathogen, at least 9 times out of 10, you're going to land on resistance, host resistance, right? So, but it's such a hard thing to accomplish. especially if you're talking about, like a host plant that's a tree that doesn't come into reproductive capability for 5-10 years, whatever, like it's so exciting to hear, at least some part of the process could become much faster with tools like this.
Yeah, but also, better. So it's not just like, even for the same speed, you would find more genes. You know what I mean? So it's faster and better is the idea.
Well, yeah, because like you said about understanding the relationship between nematode size and numbers.
Exactly.
You can,
and these are independent traits. And so you want both. You want genes in the plant that give you the smallest number of nematodes possible, and those that in fact are themselves small. Yeah. Right? 2 independent traits. You want to breed for both, for example. And so, you kind of stack all this up.
So the next question that I was going to ask you was, do you think that your work helps shed more light on nematodes as a pathogen in general? I think I can figure that out definitively. Yes. You mentioned in I watched a video of you kind of doing a summary of this research, right? And you mentioned nematodes as an orphan disease. Do you think that this, well, I guess I'll ask you to explain to my listeners what is an orphan disease, but then do you think that this makes it less of an orphan disease?
Yeah, it's interesting. So an orphan disease generally means something that is objectively important but understudied. Yeah. And so you'll get these in, neglected tropical diseases would be the kind of equivalent for humans. So there's a lot of these, and there's people that focus on those. And I think for nematodes, they're this definitely an orphan disease because they're sort of easily overlooked. They're literally overlooked, but really easily overlooked. Yeah. And so not enough people work on them. And so often when I go and give seminars in places, kind of bang the drum and, really want as many people as possible to work on these. And there is not a causal relationship between me doing that and the number of people that work on nematodes, but there are more people working on nematodes these days. Yeah. And so it is definitely a growing field, not a shrinking field, and that feels really good. So whether they are, fostered or I don't know what the analogy would be. I don't know, but definitely, exactly. But definitely there's a lot more people that work on them, and that feels really good because I think there's so much interesting biology. And it's the kind of biology, we even barely touched on it, but it's the kind of biology that really only happens in these kind of parasitic interactions. So you can't find this by looking at model nematodes, for example. And I think there's something so, it's maybe deep, or I'm not sure what it is, but there's something about, really communication between kingdoms of life is the foundation of life, right? I mean, nothing lives alone. Yeah. Everything communicates. Yeah. And so, it's a really interesting question. And these parasitic interactions or pseudoparasitic interactions can give you really interesting insights, I think, into, how that works. So I think, I would say they're less of an orphan these days than ever before.
Yeah, I loved hearing you talk about that concept because it is so interesting because, you take here in Iowa, soybean cyst nematode, the most important pathogen as far as yield loss on soybeans, right? And yet, I don't know that I'd say it's understudied per se here, but it's perhaps, I don't know, but the perception of it by farmers is pretty low because you have this thing that's coming in and it's causing yield reduction without an outward symptom, right? Like, so it's not something that they're seeing happen. So in that way, I do feel like it's orphaned, but I think that perhaps with, advances like your lab's made, like it becomes more visible, I think. I mean, literally, because of all the photos that you took, right? Like,
it's probably the most photographed plant parasite nematode by far.
Yeah. So
probably not nematode, I would imagine. But I think you're right. I mean, look, this is not a criticism of farmers, right? It is difficult to when you can't see the thing to infer that is the cause of your problem, especially when there are things you can see that are the problem, right? Especially furry things on the leaves, right? And so, so it's totally understandable. But it's work like you do about, you do more on that than I do, I think. In kind of really, spreading the message and making sure people know, that this is a problem, what to do if it's there, and all that kind of stuff. And so, I think it looks, I think it's working.
brings me right into the final one, Sebastian. So if you had to explain your work to, someone close to you that doesn't have a background in pathogens or plants even, what do you say to them, and how do you convey the importance?
I try my best not to. Yeah, I don't know. I mean, it's so hard.
But I think it's important that I think it's
so
important that people know about things like this.
Yeah, it is. It is. And I typically, you will always, you'll be familiar with this. You will always try and tailor your explanation to the knowledge of the person you're talking to, right? And their kind of backgrounds and understanding. And almost always you can find something that resonates with them about the thing. So it's really hard though. And, so sometimes I've tried to use things like analogies that work well. Some of these, though, you got to be careful, right? Well, sometimes they don't work, right? At all. But sometimes you got to be really careful. So, like, I used to use an analogy that I had some really deliberate way of saying it, that this is the wrong words but the right connotations, and that the nematode makes a cancer in the plant, like a tumor, right?
Yeah.
And so it's definitely not a tumor.
Yeah.
Okay.
Yeah.
Like,
yes.
So if you tell that to someone with a scientific background, they're not. That doesn't resonate, but it does have the right kind of connotations in, ordinary parlance. And so I don't think that's a good idea in that specific example, but, that's the kind of thing I try and do, like try and give somebody something that is a sort of, that they can identify with as, a problem. And I've started using this new organ one, and I quite like that. So
I like it too.
Good. Okay. Thank you.
So,
very kind.
With that, Sebastian, this is your time at the end. Make any acknowledgments you want, point people to any resources you've got.
Sure. I mean, it is so rare that I actually do anything in the lab. And so obviously, there's so many people that contribute to these things, and both kind of intellectually, but also technically, right? And so this is a team effort. And so, you got to give shout out to the whole teams that work on this kind of stuff, and not just, our team, but the teams that we collaborate with. So for example, the AI teams, there's just no way we could do it without them, all the way down to the poor technician who's got to pour the media plates, 10,000 plates, first day on the job. It's pretty rough. So, so many people contributed, and, it's what we see at the end and what we kind of talk about is at least the sum of all those parts together. And so, eternally grateful for their support.
Absolutely. Where can people go if they want to find out more about this?
The internet websites. I don't know if you have an opportunity to link stuff, but we can put some stuff. We'll do that. We'll make sure there's something in it.
Excellent. Well, folks, this has been a treat for me. Last week I got an email from my boss saying, "Hey, did you see this guy's coming for seminar?" I said, "No, I didn't see it, but now that I did, I'm going to reach out to him." And thankfully, Thomas Baum, I got to give him a shout out because he really helped me get a hold of you and get this set up. So thank you, Thomas. And folks, I just hope you learned a lot. I sure did. And this stuff has me so excited. So thanks for listening, folks. We'll see you next time.
I See Dead Plants bridges the gap between plant science research and the impact it has on our daily lives. It is brought to you by the Crop Protection Network and the North Central Integrated Pest Management Center, which are both partially supported by the USDA National Institute of Food and Agriculture. For more information on today's topic or other pest management issues, check out our show notes, cropprotectionnetwork.org or ncipmc.org. Remember, this information is for entertainment purposes only. The statements made in this podcast should be interpreted within the limited context of the particular topics being discussed. Contact your state extension program for local information on pest management. For the full non-discrimination statement or for accommodation inquiries, go to cropprotectionnetwork.org/about. Music written and recorded by fellow plant pathologist Sean Toporek. Thank you for listening to I See Dead Plants.